Good afternoon, viewers at home. This is another opportunity to talk to you on our lecture series of the educational series of Omega Bio-Edutainment. Today, we are furthering our discussion on genetics and the environment. Genetics, uh, genetics, and the environment. Don't forget that the last lecture we had, I told you that the first thing we are going to do is to focus our attention on genetics first. Then thereafter, we'll continue with how the environment will actually influence genetic makeup of an individual. So the last time we actually met, we actually talked on some genetical terms that are mainly used in the genetics. So today we are going to talk on the Mendel's laws. The Mendel's laws. First law, second law, and the third law. So the first law of Mendel is actually refers to as the law of dominance. Law of dominance. Okay. This law is actually stating that some alleles, just like I said, which it states that some alleles which are variants of particular gene found at the same chromosome are dominant over the other. So that is, don't forget that I told you that if you have a chromosome like this, let's say this is a chromosome, the law of dominance, if this is a tallness, if this segment is calling for tallness, so on the allele, don't forget that allele is the alternative form of this gene. So the other supposed to be shortness, that is short. So what Mendel is actually saying is that this character, capital letter T, is overshadowing the allele on the other segment of this chromosome. So that is actually regarded as the law of dominance. And basically, what Mendel's, all the characteristics of Mendel's, uh, all the law of Mendel is actually centered on the law of dominance. So, for example, if there are two contrasting characteristics or contrasting traits that cannot actually fulfill the law of dominance, that means Mendel's law will not be applicable. For example, in the case of when you cross, in the case when you cross a red pea plant, if you cross a red flowered plant with a white, if this is a white, okay, so when you cross this, you will discover that your first filial generation, all of them are going to be what? They are going to be red. They are going to be red. So, and don't forget that you always get your F2 generation by crossing the offspring of the first filial generation. That is, you want to cross R, sorry, small letter, capital letter R with capital letter R and a capital letter R, I mean small letter R, so that by the time you cross this, this is going to be RR, this is going to be RR, this is going to be RR, then this is going to be recessive RR. So what this means is that if you look at the phenotype of this is going to be red, so this is going to be white, whereas this is going to be pink. In this, so you can now see that we now have three different colors. At the end of this crossing, we have red, we have pink, and we have white. So in this case, that means the law of dominance is not in operation. because that is why we have three colors. So in case, if in any case where the law of dominance is in operation, that means you are going to have a trait and the allele, and that trait is going to be dominant over the allele, which is going to be the opposite of that. Okay, so that is that about the law of dominance. We are going to see more of this by the time we go on, but you should let it be established here that the first law of Mendel, the first law of Mendel, and all the laws that was postulated by Mendel is actually applicable to the plant or the character that they have the allele and you have the trait that is going to be. So that's why it says the law states that which are variants of a particular gene which are found on same chromosome locus are dominant over the other allele, just like it has been explained. So the second law, which we normally call the first law, but it's going to be second law as we will consider it here. Some textbooks do not actually regard the first law, which is the law of dominance, as a separate law. So, and that is why this one that I'm going to call the second law is actually being regarded as a first law. So the second law, but in particular, it is the first law, so to say, is regarded as law of segregation. The law of segregation or the law of germinal unit. What this law is actually telling us is that characters, okay, that for example, let's say you have a gamete. Let's say you have a parental gamete like this. If this is a tallness, if this is a tallness as is being applicable, don't forget I told you that if you want to do genetic crossing, you must be very, very observant that you differentiate this type of crossing from what you normally have as a multiplication sign in mathematics. Now, what this one is actually saying is that this one, this gamete is going to separate during fertilization. Is going to separate so that this one, you prick this with this. So, and you also cross this one with this so that you have this. Then this one with this, you have this. Then this one, you have this. So this one will be regarded as the first filial generation. However, that each of these, that is either small letter or capital letter, each of these will behave as a unit, as a germinal unit. So that is during the process called. So it was the first law of germinal unit, which actually brought about what is regarded as a gametogenesis. Gameto, that is from gamete. And don't forget that I told you that the genetic notation for gamete is this one is being referred to the female gamete. This is a female gamete, whereas this one is a male gamete. So what this one is telling us is that if these are the parental gamete, that is if this is a male, let's say this is a female and this is a male. What this one is telling us is that during the process of gametogenesis is a process whereby the two gametes of the two parents will come together in a process that is being called sexual fertilization. So the production, all these are called the gametes. So the production of gametes here is actually being referred to as gametogenesis. Now, just like I told you earlier on, that this kind of crossing is no longer being used because it is confusing. By the time you start crossing your using your crossing diagram, anything you have like this, it has a different genetical implication. Oh, that's why they now introduce what is now called the Punnett square. The Punnett square. So Punnett square is a graphical representation. It's a graphical representation or graphical notation that you can use in crossing terms. Okay, now let's consider this type of exactly what we have here. Let's consider it. So then don't forget that because this one is dealing with two parents, that is male and female.